Hello students, this is Dr. Henk. Today we are going to talk about scientific method or how to do science. So with this lecture we have some specific objectives. First of all, we will define what is science, and also then we'll get to know scientific method. So we'll go through all the steps, and while we are doing this, you will be actually encounter some new terms for you, and we'll try to discuss the meanings of these terms, and also make sure that you are going to read the scientific method handout after watching this, so you can actually get some reinforcement for some of these terminology. And at the very end, I'm going to be using two case studies to illustrate how the scientific method works. Okay, first of all, what do you think about science and doing science? So what are the first thing that comes to your mind when you hear this word science? So these are some latest discoveries of science. Recently, scientists have actually discovered some Earth-like planets beyond our solar system. Not just a few, like in hundreds. So the next step would be to find the life on those exoplanets. And also, bioluminescence is a really cool phenomenon in biological world. And until recently, we knew only a few fish that can actually do bioluminescence. But recently, scientists have actually discovered hundreds of ordinary fish which are capable of doing bioluminescence, but we could not see it before, but now we have methods to see it, identify this animal. This is actually a reptile, it's not a dolphin. So this is actually an extinct reptile called ichthyosaur. So this marine reptile was once dominated our seas. But how did they actually know about the colors of these organisms? And I'm pretty sure that you guys may have watched Jurassic Park and Jurassic Land, and there are dinosaurs with vivid colors, right? So when the animators were coming up with those colors, they basically used like some extant animals, some similar animals, and also their imagination. But for the first time, scientists have uncovered the pigments of ichthyosaurs, and they analyzed those pigments, melanophores, and they are convinced that ichthyosaurs were dull creatures like grayish. So that's something new. And also very recently, like 2 years ago, a team of scientists from China, they actually completely reconstructed the plumage pattern of a small bird-like dinosaur. Again, they've used the same pattern. So it's really cool. And also some people are trying to actually use organic stuff to produce renewable energy. But what is science? And this famous guy said that science is nothing more than the refinement of everyday thinking, right? And also another famous guy, he said science is a way of thinking much more than it is a body of knowledge. And in both cases, it is very clear that science is heavily involved with critical thinking. Let's go back to several thousand years back. Okay, so let's try to get the root of this word science. So sci means to know. So to define science, it's a way of obtaining knowledge about the natural world around us using certain formal procedures. And this idea was first proposed by Thales before Aristotle, but Thales didn't write much compared to Aristotle. So some people regard Thales is the first person who proposed the scientific method, and some people says it's Aristotle, but let's say both. And in this definition we talk about certain formal procedures, and collectively we call these formal procedures as scientific method. Okay, so who's doing science? Those people are scientists, right? So how would you explain your mental picture of a scientist? In other words, what is your first impression when you hear about this word scientist? So close your eyes and visualize and think about it. You got the idea, right? So when I was a kid and when I was thinking about this term scientist, I visualize someone like this: a person, a serious looking person in a white lab coat, and maybe he is using a microscope and he may handle some beakers or test tubes. So that was my imagination of a scientist. So what is the job of a scientist? So they are paid to ask interesting questions about the natural world, and the other part of their job is to actually find potential answers for these questions using scientific method. Of course, this guy in the white lab coat is trying to find an answer for his question, but personally my view of a scientist changed dramatically as I grew from a 13-year-old boy to an actual scientist. So there's another breed of scientists like this one. So I am one of those, but not an extreme field biologist like this one. So these guys are in North Pole. They are collecting blood samples from a polar bear. So their lab is the natural outside world, right? And they are dealing with one of the most dangerous animals on the planet Earth. So it's more adventurous and it's always fun to be outside. So there are several breeds of scientists. So some people love to be inside a lab and do their science, and some people like to be in the field and do their science, and some people like me do both. So I do lab work and also I do field work. Okay, so now let's get into scientific method. So it's very simple. I really want to emphasize this. It is a very simple process everyone can easily understand. So there's 3 major steps. The first one is careful observations, and I added careful because it's very important, not just observations. So when we do science, everything starts with careful observations, and if you understand what is going on, then it's solved. But if you do not understand what is going on, then you will come up with questions, and then as one of the owners of a human brain, you will always come up with speculations for this question, right? So it may be this, it may be that, and the scientific method actually gives you some opportunities to test your explanations, and you can test one explanation or you can test multiple explanations, and we are using a 7-step process to do this. Okay, so as I said, we all are doing scientific method, right? So this is the prime example. You have done it and I have done it, and my brand new son is about to start doing it. The first step, make an observation, and then if you don't understand it, then you are going to start forming hypothesis, and then you have to test your hypothesis, right? That's why you are performing your experiment, and depending on the data that you have collected from your experiment, you can analyze your data and finally you can report your findings. So again, we all have done it and we all are gonna do it. in this class too. Okay, so I tried to make it a little bit more formalized, so we can actually tackle each and every step and learn a little bit more about each and every step, and then eventually you're gonna apply this to your group project. The first step, observation. So I would say I noticed that there are trees with green tops and white flowers, and then I'm going to be wondering, that's the next step, the questioning part, right? I wonder why there are white flowers, not purple flowers. And then based on my prior knowledge and experience, I'm going to come up with a hypothesis or a potential explanation, and then I'm going to convert it to a prediction, something testable, right? So in the next step I can test my hypothesis. There are three major ways of hypothesis testing: observations, experiments, and modeling, and we will spend some time discussing about each and every step of hypothesis testing. So after hypothesis testing, either your data set will support your hypothesis or it will not support your hypothesis. If it is not supported, then you can always go back to step 3 and you can modify your hypothesis, so you can come up with an alternative hypothesis and test it again, because sometimes there are multiple answers for the same question, right? Okay, if it is supported, that's great. Then you can actually conclude your hypothesis and your work, and finally you must report your work. If you don't have any plans to report your findings, then there's no point of doing it, right? So science is all about the discovery and sharing that new knowledge. There are some scientists, so-called scientists, who are doing science and research, but they never share their findings. So what's the point of doing that science? So I want you to be a good scientist who do science and share your discoveries with other people. Okay, so let's talk more about these specific steps of scientific method. So we'll start with observations. Okay, so in any science, observations are the root cause and the most important first step, because if you observe the natural world with a curious mind, you will come up with questions, and you can directly observe things using your naked eyes, your 5 senses, and sometimes you can observe using published works. So basically, read more. When you read more, you will come up with more questions, and also sometimes you can do dynamic models to generate questions. So all of these methods which are going to generate questions are methods of observations, right? So let's discuss about some examples. Okay, so I'm gonna come up with 3 observations. So my first observation is Florida panthers have a very low pup survival, and this is Florida panther. It's one of the large carnivores in United States, and there's a landlocked population in Florida, and it's an endangered species. But the sad story is their population is keep declining, mainly because of very low pup survival. And also we have observed that males live longer than females, again an observation, right? And kangaroo rats are nocturnal. We don't see kangaroo rats during the daytime. You have to go to their potential habitat at night to see them. They are active only during night. Okay, so now let's move to the step number 2, which is questioning part, right? Then you are going to be wondering about these observations. So if you do good observations, it will lead to ask good, interesting questions. And again, I want to emphasize the importance of quality observations. The depth and breadth of the questions depend on the quality and the detail of the observations. So that's why we are actually going to use a field notebook to summarize our observations. Sometimes you may think, okay, so I can just do my observations and remember everything. There are some issues with that. I do not trust my memory. I'd rather trust a note, a written note. So when you do observations for this class, you must take notes. So by that way, we can guarantee that you are going to collect quality and detailed observations, which is going to lead interesting questions. And by the way, this is a kangaroo rat. It's a really cute animal, and typically they are distributed in the southwestern US, mainly in very arid areas, in deserts. Okay, so how are we going to convert our observations into some potential questions? Okay, so first our observation was that Florida panthers have very low pup survival. Now I'm questioning why. Why do Florida panthers have a very low pup survival? And again, why do males live longer than females? And why do kangaroo rats are nocturnal? Do you remember the next step? Step number 3. Yes, it's hypothesis generating. So hypothesis is a possible answer based on your previous experience or your previous knowledge, and it's actually an educated guess, not just a guess, an educated guess. You need your prior knowledge to add into this educated part, right? So that's why you need to gain knowledge of your discipline before you do your science. So before you do an experiment or a study, you have to extensively read about the previous work. Then you can always come up with good hypotheses. So hypotheses are formulated based on several things. So you have to think about your observation, of course, that's the basis. And then you can ask many questions to tackle the same observation, right? So you have to be very careful with your questions. If you ask the right question, then you can come up with the right hypothesis. And of course, you need your prior knowledge when you are formulating your hypothesis. Okay, so let's make some hypotheses. Okay, so in the case of Florida panther, so we are going to say Florida panthers have a very low pup survival because, and now I'm going to add my educated guess. I've been reading about these guys, so I'm going to say because they are genetically inbred. So it's a very small population. When it comes to small populations, you are going to be basically mating with your siblings, right, which are genetically almost identical to you, and that will eventually deteriorate your genetic diversity, and we call it genetically inbred populations. Okay, so the fourth step is coming up with predictions. So in this case, the most important thing is we are going to consider that our hypothesis is true when we are making our predictions. So it's a possible outcome determined before the hypothesis has been tested, right? And again, when we are making our predictions, we will consider that our hypothesis is true. Okay, so let's convert our hypothesis into prediction. Now I'm going to stick with the Florida panther just to keep it straightforward. So our hypothesis was Florida panthers have a very low pup survival because they are genetically inbred. But how are you going to measure this genetic inbreeding? So one of the ways to measure it is to measure the genetic diversity. So I'm going to predict that Florida panthers have no genetic diversity, or it may be very low genetic diversity. The next step, it's going to be step number 5, hypothesis testing. So once you have your hypothesis and once you are ready with your predictions, you have to test the validity of your predictions by hypothesis testing. So during this step, you have to do specific things. First, you have to collect data. You have to collect right data which is actually relevant to your question, your hypothesis and prediction. And then you will analyze your data. Typically, we'll do some statistical analysis here, and then based on the output of the statistical analysis, then you can make some inferences. There are 3 major ways of doing hypothesis testing in these days. The first one, observations of patterns. So this is one of the fundamental ways of hypothesis testing. And the second one would be manipulative experiments. This is the gold standard of hypothesis testing. And also you can do dynamic models to test hypothesis. So we will discuss more about these major steps or major methods of hypothesis testing once we are done with our discussion about the steps of scientific method. Okay, so we are at step number 5, right? Okay, so after you are done with step number 5, your statistical analysis may support your hypothesis. Then that's great, so you can conclude your study. But sometimes it may not be the case. So if your statistical inferences are not supporting your hypothesis, then you have to go back to the step 3, and you may need to modify your hypothesis, or you may have to come up with a different hypothesis and redo this process. So that's the beauty of scientific method. It's an iterative process, right? And it's very specific, and anybody can do it, and you can repeat it. So okay, let's move to the next conclusion. So if it is actually predicting our results correct, then we are done. So let's conclude, and then we can start writing our project, right? And if it is not the case, we may be wrong, so we can modify the proposed explanation, or we can just move on. Are we done? Do you think that we are done? Yes, one last step, otherwise you won't be famous, so there's no point of doing science. And you know that this story, right? This is someone who discovered the most important concept in biology. And this guy discovered the same concept independent of guy number 1, Charles Darwin and Alfred Russel Wallace. So everybody talks about Charles Darwin because he published, or he shared his findings with the world, but Wallace was late, so nobody talks about Wallace. So this is Darwin's big book, The Origin of Species. This is the book that he actually introduced his ideas about a potential mechanism to explain evolution, the natural selection, right? And I tried actually reading this book several times, and I hate to say this, but it's very boring. That Victorian style, I just can't stand that, because it takes maybe 10 pages to say something that I can write using a single sentence. But that was the style at that age. I'm gonna attempt to read this book again, and if you like the Victorian style, please read this book. It's a really okay. So the last step is reporting your findings. There are various ways to do it. So way back when you discovered something new, you would typically report it to your regional leader. So when you find new land, you go and report it to your king, right? And nowadays we do something else. We do different things. So you can actually go to professional meetings and give oral presentations to share your science. And also, as scientists, we publish our work, we write papers, and also some people, they write books to share their findings. And you can do your science and you can share your findings too. So you can actually do poster presentations at regional meetings, in professional meetings. And we are going to be doing some reporting in this class too. So at the very end, you are going to be doing an oral presentation to share your findings with the Longwood people and some Farmville community. And you may recognize some of these people, right? So this is one of my PRISM students, Connor Perry. He's actually presenting his PRISM research at the end of summer of 2016. So they did some really cool studies to look at the impacts of invasive crayfish on native crayfish. And this is Dr. Fortino asking, looks like a hard question. Okay, so that concludes our discussion of the scientific method, and we discussed about all 7 steps. Now we are going to be allocating some time to talk about hypothesis testing. Okay, so the first method of hypothesis testing is observations of patterns, and I must add extensive observations of patterns. And typically we do observations when the experimentation is difficult or unethical. So for an example, think about Florida panthers, right? They are an endangered species. The population is less than about 50 animals. So it's very difficult to actually pull out some of those. And also it is not ethical to pull out some of those animals and do an experiment. And also think about invasive species. So do you think it's ethical to study an invasive species after introducing it to an uninvaded habitat? So basically I'm creating my own question, right? So I'm introducing an invasive species to an uninvaded habitat, and then I'm going to start researching on them. That's not ethical. So for ethical and some other reasons, we cannot do experiments all the time. So here comes observations. So you can always collect data by observing patterns. You can observe animals to study their behavior. You can actually observe and count organisms, and you can study their population dynamics. And then you can compare this data with a control group, right? And this is actually the naturalist approach, and we discussed about the natural history at the very beginning of the class. So when the natural history was developing in like early 1600s, they basically just observed plants and animals, their form and function, their behavior, and they actually kept notes about those, and then they tried to actually recognize major patterns. And the classical ecology is largely developed based on observations too. Okay, so now we are going to start discussing about a case study to illustrate how are we going to do some observational studies. So again, we can do any experiments on Florida panthers, right? Because it's an endangered species and there's only a few species, few individuals. So this species is once a widespread carnivore. So nowadays you can actually find Florida panthers, or sometimes people call mountain lions, cougars, or pumas. It's the same species. They are widely distributed in western US, Central America, and South America. And several centuries ago, they were all over the United States and most of the Canada. But you know what happened? So early settlers, they started spreading from East Coast, and they killed most of these large carnivores, right? But there's a landlocked population right here in Florida. So currently we are classified it as a subspecies, and we call it Florida panther. So in 1970s, there were only 6 individuals, and scientists who are studying these large carnivores, they predicted that this really cool large carnivore will be gone in 25 to 60 years. So they listed it as an endangered species, and then they actually started the recovery program. Okay, so we are going back to our one of our original observations, and we are going to be actually elaborating on this. Our observation. Okay, so Florida panthers have a very low pup survival, right? And then we asked this question because we didn't know the answer. Why do Florida panthers have a very low pup survival? And then we converted it to a hypothesis. Florida panthers have a very low pup survival because they are genetically inbred. And finally, to test our hypothesis, we converted the hypothesis into a prediction. Florida panthers have no genetic diversity. Okay, so how are you gonna collect data on genetic diversity? This is how they did. So what they did was they trapped a nice sample of Florida panthers, and first they actually look for any signs for low genetic diversity. So sometimes if you are inbred, there are some external signs of genetic inbreeding, and also they collected some blood samples. So with these blood samples they can actually get DNA extracted, and then they can actually try to measure the genetic diversity. So when they were actually studying the morphological evidence or external features for inbreeding, they found that many Florida panthers have signs of suppressed immunity, mange or ringworm issues, and they collected this data mainly by game cameras. And also almost all individuals that they've handled had kinked tails, and many of them had cowlicks too. And originally the biologist who described this panther as a subspecies mentioned that this kink-tailed and cowlicks are specific characteristics of this subspecies. But now we know that it's not the case. These are signs of inbreeding, and you can find these signs in some other animals too. Maybe some of your dogs have these cowlicks and kinked tails. These are signs of inbreeding. And then they did some genetic analysis based on their blood samples, and they found that there's only several genotypes. In this case it's color coded, right? So green, yellow, and a little bit of gray. So only 3 genotypes in the Florida panther populations. This is not good. It's an indication of very low genetic diversity of this population. So if you want to find a solution, or if you want to reverse this low genetic diversity, you can do some things, right? And we discussed about one of those in chapter 2. Do you remember? Genetic rescue. So basically you can actually introduce animals from a distant population with rich genetic diversity. So in this case what they did was they actually introduced 8 panthers from Texas. That's the nearest population, but the Texas panthers are pretty good with their genetic diversity. So after several years, this is what they have observed. So what do you think? Do you think that the genetic rescue worked with these Florida panthers? Of course, now they have several genotypes: green, yellow, blue, red, and gray. So 5 genotypes is way better than only 3. right? So in this case, they augmented the population with some new genetic material to boost their genetic diversity. So this is a really cool study, and they did most of their research using observational studies. Okay, now we are going to be talking about experiments. This is the most powerful procedure to test hypothesis, because you are going to be doing it under controlled conditions, and you can actually tease one factor at a time. You are going to control all the factors except one, so you can actually look at the impact of that specific factor on your study subjects. And again, this is the gold standard of hypothesis testing. So when we are doing experiments, we try to keep all factors relatively constant except the factor of interest. And when we are actually doing experiments, you have to do something called experimental design. So you have to think about how to treat your groups and what are your control groups, and also you have to think about what kind of statistical analysis that you are going to be using. So when we talk about experiments, as I said before, some people do lab experiments. So these are some of my Prism students doing some lab experiments on crayfish, and also we do field experiments, right? So here my group again doing some field work. And sometimes you can manipulate your experiment on a spatial scale. So sometimes you can do your experiment inside a flask, and we call them microcosm experiment. So it's a very small system. These are good for like to get some first thoughts about your question, but it's really hard to extrapolate findings of these microcosms to the real world, because think about the scale. You really cannot consider this flask as the real world. The scale is very important, and sometimes ecologists do ecosystem scale experiments. So in this case, you can see they basically cleared an entire watershed to see the impact of logging. This is the best way to do it, but is it ethical? And it's a sad story. But typically those scientists, they actually team up with logging corporations. They are going to log this area anyways. So then they'll talk to those logging corporations and they come up with a deal. So they both can do their jobs. So the scientists can study the impacts of logging, and the logging corporation can get their timber. And there are some other ways that you can merge both worlds. Those are mesocosm experiments. So basically you are going to create an ecosystem in a reasonable sized container, and these are my students. Again, we are doing some experiments on crayfish. We were looking at the impacts of invasive crayfish on natives, and we use these large 360 gallon cattle tanks to create ecosystems. And again, you can replicate your system with mesocosm experiments, and the data that you are going to generate is going to be more realistic than lab experiments, and we can talk more about this later. At the same time, please read your handout, scientific method handout, and you can actually get more information about experiments and some of the terminology. Okay, so our second case study to illustrate how experiments are used to tackle some hypothesis testing. So this is what I have done for my PhD research. I basically lived in the Mojave Desert in the middle of nowhere for 2.5 years to study these two fish. So this one, Mojave tuishab, it's an endangered fish in California, and there's only 4 populations for the entire world. But in one of those populations, Lake Chwende, we have observed western mosquito fish. So somehow they get into the lake, and it's an invasive species, and there are many horror stories about mosquito fish actually dissipating native species after the introduction. So we were so worried. So my task was to look at the potential impact of western mosquito fish on this endangered fish. Okay, so let's try to formulate our proper sequence. So my observation: a population decline of endangered Mojave tuichab in Lake Chwende, right? So then I'm gonna ask the question, why do endangered Mojave tuichab populations decline? Then, based on my previous knowledge, I hypothesized that Mojave tuichab populations declined because of low larval production, so reduced breeding. So how to test that? So my prediction was invasive mosquito fish prey upon Mojave tuichab larva, right? So this actually allowed me to design a really simple and clean experiment. Again, we use mesocosms, those large cattle tanks. So we established two groups of cattle tanks, and for each cattle tank I stocked some tuichubs. So in this case 8 tuichubs. This is the endangered fish, so I stocked 8 tuichubs in each tank. And for one group I stocked some mosquito fish too. So in this case my treatment is Mojave tuichab plus mosquito fish group, because I'm introducing my treatment mosquito fish, and I kept the other group without any mosquito fish. So this is my control. So when I'm analyzing my data, I can use this control as the basis, because there shouldn't be any impacts of mosquito fish in this control group, right? So the treatment is mosquito fish and my control group is the tuichubs only. And so I mentioned about two groups, right? And you just can't do experiments with just one tank, right? One tank of tuichub only and one tank of tuichub and mosquito fish. You need to replicate your units, because there's always some variation of biological systems, right? So the typical standard is 30 replicates, but for this research, no, there's no way that I'm gonna maintain 60 tanks in the middle of nowhere. So what we did was we replicated each 10 times. So I had 10 Mojave tuichub only tanks and 10 Mojave tuichub and mosquito fish tank. And you may wonder whether why I have depicted like two different fish here. So mosquito fish are highly sexually dimorphic. So this one, the lower one, is a male and the upper one is a female. So the females are much larger than males. And also I stocked 25 males and 50 females just to balance the natural sex ratio of this fish, so I can expect more realistic data, right? And my experimental unit is the fish populations in each tank, right? And please read more about treatments, controls, replicates, things like that in your handout. Okay, so these are some of my results. So what do you think? So we actually stocked fish and then we maintained those tanks for 3 months. So during that period, both species, both endangered tuichubs and invasive mosquito fish, bred in the tanks. So after 3 months, I actually harvested all the species, all the fish in both groups. and this is some of the information. So in this graph you see the numbers of Tuishab larvae. This is the reproduction of the endangered species, the progeny of the endangered species. Two groups. In my controls, on average I have about 34 Tuishab larvae in each tank. But when there's mosquito fish, look at this, it's about 5 Tuishab larvae in each tank. So this clearly shows that when there's mosquito fish, there's a negative impact of mosquito fish on endangered Tuishab reproduction. And this may be because of predation of mosquito fish on larval Tuishabs or Tuishab eggs, or it may be competition, right? But I mean, it's really cool that you can generate more realistic data when you are doing mesocosm experiments. But in between stocking and harvesting, it's like a black box. You really cannot see what's going on. So I did some follow-up lab experiments to get more insights on this, but for our purpose, these are our results. So it's very clean, so you can make conclusions, right? Okay, so the final method of hypothesis testing is modeling. So typically a model is a simplification of something, right? So it's a generalized or a simplified natural phenomenon. And we can use these models to predict the outcomes under different scenarios. And always you need to use mathematical algorithms and computational power to run models. And some examples are weather models. So we hear the predictions of weatherman every day, and they are doing it based on some weather models, right? And in ecology we are using lots of models, but I'm gonna stick with one kind of model to explain the dynamic modeling in ecology. So that is ecological niche modeling. So in this case, first we have to collect some data, right? So we would collect some data on, let's say, the distribution of a given species, right? So species field observations. So first we have to collect some data from the field. The red dots are the presence and the X marks are absences. And also then we have to combine this species distribution data with some environmental data, because our goal is to actually predict the range, the future range of this species under different scenario. So we may collect the data on the rainfall, or we may collect data on the vegetation cover, or we may collect data on temperature or humidity. And then we are going to link each data point and then feed it to our model, right? So we are going to run a bunch of mathematical algorithms, and then we'll generate these response curves. Based on these response curves, then we can actually predict some spatial outcomes. So for an example, what is going to be the distribution of this species if the temperature of that region has changed by 2 Celsius, right? Or we can predict what's going to happen to this distribution of this species if the rainfall of this region dropped by 100 millimeters. Or we can predict something like what's going to happen to this species distribution if an invasive species invaded this habitat. So this is a very useful tool in ecology because it's going to give us the power to predict future scenarios. Okay, so let me show you an example for ecological niche modeling. So you may know about Audubon Society, right? This is one of the oldest citizen groups in the world. and they are specifically dedicated to conserve birds in North America. So recently they have ran some niche models to look at the impact of climate change on birds. So they actually did this analysis for 314 species of birds. So I'm going to show you just two. So cerulean warbler is a tiny bird. It's a neotropical migrant, and they typically migrate to United States during the summer to breed. And most of these guys are actually in Appalachia, in Virginia. So based on their niche model, they have predicted by 2080, 98% of this summer range will be shifted away. So basically, their summer range will shift north. And I'm going to show you their prediction. Okay, so look at this map and focus on this eastern US. And you see this yellowish boundary over here across Virginia and parts of North Carolina. So this is the current summer range of this species. It has a very restricted range, right? But this is 2000. But now do you see this cloud, this yellow cloud moving north? Look at this. So 2020, so this is 2020 range. You see it? And this is 2050, further going north, but also the intensity is dropping. By 2080, the species is hardly occurring in United States because it's basically moving poleward, just tracking the temperature. So it's a very sad story, right? Okay, so let's see what's going to happen to our national bird, the symbol of North America, of United States. So only 26% of its current summer range will remain by 2080. And again, most of these guys are moving north. So let's have a look. Okay, look at this. So blue is the winter range, green is both seasons, and the yellow is the summer range. So currently in most regions of United States, it's a winter bird, right? Because they actually move south during winter to get more food. But most of them actually breed up in north, right? Okay, so let's see. Basically the same thing is happening. We are getting fewer and fewer winter birds because the climate is going to be warmer in north, so they can actually stay there. Okay, so this is 2000, this is 2020. Now you see some white patches in Texas and some other southern states. 2050, more white. 2080, right? So basically their distribution is moving northward. So that's how you actually apply ecological niche modeling. First you get the information about the current distribution of the species, and then you'll combine it with a bunch of other ecological parameters like temperature, rainfall, vegetation, and then you can actually run these ecological niche models, and then you can predict future scenarios, future distribution under different scenarios. Okay, so this will conclude our discussion about scientific method. So we have discussed about all the steps and also various methods of hypothesis testing, and we will actually follow this scientific method very closely when we are completing your group project.